All right, we are going over the chapter of respiratory system, and we will discuss general ideas about the physiology of breathing. Here's the outline. First, we'll look at some of the components and the structures that are involved in our breathing that is related to anatomy, type of muscles, and the trachea, etc. Then we will take a look at the mechanism of breathing, pressure and volume relationship. We will take a look at physical properties of the lung, including lung compliance, elasticity, surface tension. We will take a look at how the gas exchange in the lungs takes place. Of course, diffusion is part of it, but we will go a little bit more detail of the gas transport as well as exchanging both oxygen and carbon dioxide. We will take a look at how does the acid and base balances occurs, principle of homeostasis. And finally, we will look at how do we control and monitor and regulate breathing. For example, why is it that when you're exercising, your breathing rate goes up? When you're at rest, your breathing rate goes down. Or right after the exercise, you're resting, your breathing is going down. So we will take a look at those ideas as well. All right. That's pretty much what we wanted to do. And this is pretty much the outline of what we're going to be talking about. So let's just start off with some of the structures that are related to the breathing. So for that, why don't we just go ahead and take a look at the lungs, and maybe we can, hmm, uh, here is the lungs, but a lot of things doesn't show in this slide. It just shows a lot of problems. So I guess I have to maybe include another picture, but that's okay. Uh, One of the important areas in the lungs that you probably need to know is the muscles that are involved. Okay, so we have the lungs. All right. Right here, there is a muscle, dome-shaped muscle that separates the abdominal cavity from the thoracic cavity, and that was the diaphragm, if you remember. Uh, they are encased by the chest and the rib cage. So you have the rib cage in here. This is the sternum, and so on. And this is the ribs, and so on. If you recall other muscles that were involved in here, one of which was between the ribs, all right? And these were the intercostal muscles, internal and external intercostal muscles. Those are important. On top of that, you had pectoralis muscle that was, fan-shaped muscles, if you recall, all right, and also pectoralis minor. So all of those respiratory muscles is important because if you don't have the muscles, or the muscles, you have a spasm, or you have a diaphragm is not working properly, and so on, that is going to interfere with the normal rate of the lungs. Okay, so that's one of the things that are the structures that are involved. The next structure that is involved happen to be uh, the actual trachea. So we go ahead and take a look at the actual trachea. So we can just kind of like erase some of these, and you could go and take a look at the better picture, of course, from the internet or your book. All right. So we go ahead and take a look at the trachea. Uh, it's important to know a little bit about the trachea. So I will expect you guys to know this, but the trachea or the windpipe consists of this cartilage. These were the hyaline cartilage. It was kind of like a C-shaped structure you observe. The other structure happened to be the bronchi. And again, in anatomy, you learn the primary, the secondary, the tertiary bronchi. All right. So basically, the branch is getting smaller and smaller. After that, you have the bronchioles. Bronchioles are sub branches. Again, inside of the bronchiole, there are tiny little tubes in there. So the air flow as it goes from the oral cavity passes the throat area, the pharynx, and then the trachea, and then the several different bronchi, all right? And then through the bronchioles. So if we go ahead and unwind the bronchioles, they still have the diameter is getting smaller, but they still have the airways. And then that was reaching the alveoli. And in the alveoli, these were the air sacs that is a lot of surface area. And these alveoli were covered by a lot of capillaries surrounding it, and the gas exchange happens there. So, in other words, the oxygen that you breathe in and going into from the oral cavity or the nasal cavity through the pharynx, which is the throat, to the trachea, to the bronchi, to the bronchioles, and finally, you'll have the alveoli. If I went too fast for you, you could always pause the video or also go back and review these structures for the saving time. I'm not going to label and write every single thing because this supposed you're supposed to have all that during your anatomy anyways. So these are the air sacs or alveoli. Alveoli is the area where the gas exchange taking place. And again, they are surrounded by a lot of blood vessels. These blood vessels are capillaries. So therefore, the oxygen will diffuse inside of these capillaries. And this is called oxygenation of the blood. On the other hand, they contain a high amount of carbon dioxide. So the carbon dioxide is going to go from these capillary back into the same areas. In other words, you can go upwards and so on. All right. So this kind of like resembles an inverted tree. All right. And interestingly enough, the structures in the trachea, as you know, there are mucus lining, there are ciliated epithelial to trap the dust particles in these areas and so on. So uh, those are the some of the important structures. So when the dust particle comes in and travel, let's say it's windy, it's there's a lot of dust floating around, or you're cleaning, or whatever the case might be, a lot of these dust particles, they get trapped. These cilias, they distribute what's known as a mucus. All right. So if we go ahead and take a look at the mucus lining, they distribute this mucus lining by movement of these cilia. It distribute the mucus lining. So when the dust particles are coming, all right, they will be able to get trapped, a lot of them. So in a way, is cleaning the lungs for you. Now, the air actually enters through the nose, and inside of the nose, so we're looking at the pathway of inhaled air. All right, pathway of inhaled air. All right, so here's a better picture. So the air will go into the either oral cavity or the nasal cavity. We're looking at the pathway of the inhaled air. As you inhale the air, this is what will happen. All right, so you have a lot of hair inside of the nose. These hairs would trap dust particles, things that are unwanted to enter the lungs. Obviously, that's what happens. All right. You also humidify the air and make the air warm. Okay. Now, the air will continue going towards the pharynx, which is the throat area, and then enter the trachea. You can see the hyaline cartilage. Hyaline cartilage ensures that this airway is always open. The reason why, because you want the flow of air in and out. Okay, so that's really, really important. All right. Now, interestingly enough, when you see a lot of predatory cats, they always aim for the trachea. If they wanted to catch and hunt the deers, for example, they try to aim the trachea, because once they squeeze that part, they can suffocate their prey pretty quick. All right. And then they can have a feast afterwards. All right. So if we go ahead and take a look at inside of the trachea, all right, and that's what you've done in the anatomy, by the way, all right, you'll see this is a transverse section that is cut from the trachea, all right, and you could see it says ciliated, columnar, epithelial, pseudostratified. So again, these are ciliated, means they have tiny little hairs, which is not shown because it's microscopic. All right. So, but the point is that when the substances, pollutants, pollution, dust particles, things like that, a lot of them can actually get trapped right over here. All right. And not going further down. Right. So your air, it's kind of like semi getting clean. So right over here, you could see the bronchiole leading to the alveoli that I just mentioned, and alveoli are the air sacs. These in blue color are the different type of the bronchi, primary, secondary, tertiary, and then it gets to the bronchiole. All right. Now, when we're looking at uh, these bronchiole, they actually can constrict and dilate. Now, same principle as we discussed the blood vessels, all right? Constriction is going to reduce the airflow. Dilation is going to increase the airflow. So think about the time that you need to dilate or constrict. Obviously, when you are exercising, you need more air, so dilation. When you're at rest, you probably need to bring in so much air inside, constriction. Again, you bring in more air because your muscles require more oxygen, all right, to generate the ATP that we discussed, all right? So everything comes together now, all right? The things that you learn about the mitochondria, the ATP, what do we need to do, glycolysis, aerobic respiration, all of this is supposed to be coming together now, all right? So those are the some of the ideas. Now let's go ahead and take a look at the. You can see the capillaries surrounding the alveoli, and there are some elastic fibers and a smooth muscle for the sake of expansion and constriction and things in that nature. As you know, the elastic fibers, they have recoiling capability, so when the lungs expand, it helps it to go back to original way. And again, the smooth muscles can constrict and contract and relax as well. So you'll have some ideas behind that. And obviously, the most important things are the capillary in this case, because the capillaries are really, really thin. So it allows the gas exchange to take place in here, not anywhere else. So sometimes this is known as, respiratory, or the site of respiratory, or respiratory zone, and things in that nature. All right. So that was just a little bit of background to give you an idea about, how and what these cells look like. Let's go to the alveoli. All right. And the alveoli, interestingly enough, they have a cell known as a surfactant cell. The surfactant cells reduces the surface tension, so it allows the lung to expand. This is important. It's kind of like a balloon. If the lung can expand, you could put in more air in it. All right. And obviously, during the expiration, opposite will happen. The lung is going to collapse or go back to original way. So these surfactants are really, really important. Children that are born without this surfactant, or not much surfactants are being produced by these cells, the lungs still collapse, and they have to be very careful to administer oxygen, because very soon the baby is going to suffocate and die if the lung is not working. All right. So the lung has to get working. It has to have this surfactant and so on. Now, as we grow older, the less and less surfactant is being released, and therefore we lose that capability of expansion. So the lungs of the individual that are older is probably not going to be able to expand as much. We all see some macrophages as well. These macrophages supposed to, engulf the invaders, microbes, things that are still unwanted, and somehow they wind up in here. And for the most part, they do their job pretty well. Now you might say, what about the coronavirus? What about the other respiratory infections and so on? Yes, at times they cannot keep up. The rate of reproducing of these microorganisms is far, far beyond their capability. Is kind of like having many people rioting and I don't know, taking things from the stores and looting and things like that, and there's not enough police officers to do much about it. All right, it's kind of like that. All right, so the battle is going to be lost. All right, or it would take longer for your body to get the order that it requires. Okay, so that's the idea behind what kind of a cells you can see on the alveoli. Now, let's go ahead and take a look at the Boyle's law. Again, if we go back and take a look, there is an important muscle known as a diaphragm, okay, underneath the lungs, all right? And there is a lot of relationship between the volume and the pressure. So pay attention on it and you will get this. In general, ideas is that you have the pressure always goes from high to low. The idea is that if you have a high atmospheric pressure, so let's say 760 millimeter Hg, and you have a lower atmospheric pressure inside of the lungs, the pressure goes from high to low. So that's the order in which they go. But during at times the pressure might increase in here, and as a result of that, this might be more than the pressure outside, so it's going to go opposite end. So this is where the inhalation and exhalation comes in. So let's go ahead and take a look at perhaps maybe how the inhalation and exhalation occurs, which is related to the Boyle's law. What is Boyle's law? Boyle's law indicates that when the pressure is getting higher, the volume is getting lower, and when the pressure is lower, the volume is getting higher. So they have opposite relationship with one another. Now, do not mix this with the idea of, oh, well, I thought like, in your blood vessels, you said higher volume, that gives us high blood pressure. Then how come in here, the pressure and the volume is the opposite relationship? It's because this diameter didn't change, folks. This diameter did not change in here. But when we're looking at the lungs inside the chamber is changing. So let's take a look at what is happening. Now, during inhalation, this is also known as inspiration. What happens? The diaphragm moves inferiorly, all right? During the contraction, the diaphragm goes inferiorly. So what that means, if this is the diaphragm right over here, it moves downwards. If it moves downwards, that gives opportunity for the lungs to expand. It has more room now, and that's what happens. Now, if you are sitting and listening to this lecture, I want you to put your two hands on your chest, all right? Place it on your chest and take a deepest breath that you could take. Breathe as much as you can. Feel your hands, what's happening. You can feel like your lungs is expanding, right? All right, so that's what happened. Rib cage is expanding because the lungs inside are expanding. So again, a lot of these intercostal muscles are working, and they are also helping in order for the lung to expand, create more rooms, because the diaphragm is moving downwards. So in this case, the thoracic volume is going to be increased in here, you see. increasing thoracic volume. Inside of it, there is more gas because the oxygen went in already, right? So thoracic volume is increasing. Lung is stretching, and interpulmonary volume is increasing. As a result of that, the pressure is decreasing now. The pressure is going to be lower in this case. It's kind of like almost like negative. All right, outside pressure is higher. Outside pressure is 760 millimeter Hg. You've just created a vacuum. It's kind of like a vacuum. All right. So now the pressure is going to go from high to low, and the oxygen is going to move in. And this is the idea of the inspiration. Now, during expiration or exhalation, opposite will happen. All right. So, in other words, the diaphragm moves upwards superiorly as it relaxes. All right. So in this case, the thoracic volume is going to be decreased now. All right. A lot of those inspiratory muscles, they start relaxing. The diaphragm rising, the rib cage descend. All right. Due to the costal cartilage. All right. And all of those. So your lung initially was expanded like here, they were expanded, right? The diaphragm was moving downwards. This is the opposite, all right? So now the diaphragm is moving upwards, all right? And the muscles and such are relaxing. And as a result of that, what is happening? Take a look at the volume inside now. The thoracic volume is going to be lower, but it's kind of like but I'm sorry, the volume is lower, V, but the pressure is going to be higher, and this is forcing the air to exit. In order to understand the pressure and the volume, you could think about you have a beach ball, all right? Here's your beach ball, all right? And I don't know, usually like, some patterns on it, right? So if there is a valve in here, but if you open the valve and push, let's say it's time to go home from the beach and you're pushing on the side to get the air out. When the air is going out, obviously there's going to be lower volume, but your hands is squeezing here. It's kind of like what is happening here. All right. So in other words, what happens? Well, you increasing the pressure. So the pressure, forcing the air to go out, and that's what exactly happening. And this pressure and volume relationship are known as Boyle's law. All right. So that's one of the things that you wanted to think about. All right, the next thing that you wanted to know is the physical properties of the lung. And when we say physical properties of the lung, we are talking about lung compliance. Basically, when the lung is capable of expanding normally or a lot, we call it high lung compliance. And if they cannot, then we call it the low lung compliance. Think about as you have a nice new balloon, and with a little bit of blowing air, the balloon is capable of expanding. But the same balloon that you obtain, maybe from an old, maybe 5 years old or whatever, it's the same balloon, but when you put in the air, it doesn't expand as much, and you're pushing more air. All right, you're having difficulty to blowing it. All right, so this is a low lung compliance. This is a high lung compliance. The high lung compliance are healthier lungs. All right, and the low lung compliance are basically the lungs that are not expanding well. All right, so another physical property that is important is called elasticity. Elasticity is ability to, recoil. Think about it as a rubber band. When it is you let go, it goes back to the normal, and so on. So elasticity, it helps to recoil capabilities and all that. And so as the surface tension that we discussed, the idea of the surfactant, all right? So sometimes you have a deterioration of these elastic fibers due to old age, due to oxidation, due to variety of the case might be. Like we grow older, that's what happens. Surfactant. Surfactant, when it's reduced, you get the low lung compliance, all right? Because you have less surface tension, all right? Less surface tension, lungs cannot expand as much, all right? Well, the lower the surface tension, the lung can expand more, but what it does, it interfere with the surface tension. So it doesn't expand. So that is one problem that we can talk about. In general, there are two kinds of disorders that you wanted to kind of like look at. One of these disorders is known as restrictive disorders. Uh, Restrictive disorder, and the other one is obstructive disorder. But let's say restrictive disorder, because it's related now, those are the expansion. The lungs is not able to expand as much, so there are expansion problems. All right, the lungs that cannot expand as much. All right, those are restrictive disorders. All right, so let's say for whatever reason the the lungs is you know punctured, the lung it has a knife wound, etc. You can see what happens is kind of like pushing a nail into a balloon. Now it collapses, it doesn't expand. So that would be a good example of restrictive disorder, right? All right. So whatever you can think of that the reason why the lungs does not get uh, you know expanded, those would be the restrictive diseases. All right. So let's go ahead and take a look. One of those would be deformity of the thorax. Deformities of thorax. Which means what? Which means like, your thorax doesn't have the right shape and the structure. A lot of these has something to do with your vertebrae, due to, I don't know if you remember your anatomy or not, but kyphosis, lordosis, scoliosis, those were the deformities of the vertebrae. And that would create problems for the lungs to expand normally. So in order to understand this, what I want you guys to do right now, as you're sitting, I wanted you to be kind of like a slouch forward, like, you create a hunch back for yourself, all right? And while in that position while you're in that position, I want you to, don't change your position, you're still slouching, all right? And you have exaggerated and you're pretending like, you have a hunchback, all right? Now I want you to inhale as much as you can. You'll notice like your lungs is not expanding as much. Again, you could place your hands on your chest while you're doing that and feel it. And you notice you have a difficulty breathing too. All right. And that is because the lungs is not expanding. All right. So that can happen. Another problem of restrictive disorder, there could be ossification of the costal cartilage. All right. So you have the ribs. And if you remember, over here you had costal cartilage, right? And over here you had the ribs, right? So these were the costal cartilage. If you recall, this side, I'll do a better job. Now, in reality, they have more ribs, but I'm just showing you. So this wiggly line right here, these are the costal cartilage. Right? That's what they are. Costal cartilage supposed to be flexible. So when you have the lungs that is inside, the lungs, as they are expanding, when you are breathing in, what happens? The lungs is expanding due to this cartilage being flexible. The ribs and the lungs also, all of those are expanding. Okay, but when you have mm, when these costal cartilage are not as flexible, lungs is having a hard time to expand. Now, there may not be any problems with your lung in terms of expansion, but due to restrictions on the costal cartilage stiffness, they're unable to expand. And that is the problem. All right. So another expansion problem could be paralysis of intercostal muscle. And the paralysis of the intercostal muscles, if you recall, the intercostal muscles were between the ribs, right? So when you have these muscles paralyzed for whatever reason, all right, maybe lack of oxygen, maybe, other complications, but again, they are not going to be able to expand as much, and as a result of that, it will affect the lung condition. All right. So those are some of the ideas. To further understand, you can think about the inflammation sometimes that is occurs in our lungs. You can think about perhaps maybe the some kind of infection or fibrosis, all of those, when the scar tissues replacing normal lung tissues, usually we call this a fibrosis, all right? Some lung complication, tuberculosis, chronic inflammation, those type of things could cause. And as a result of that, obviously, is going to diminish the lung compliance we discussed. All right. So very important to understand these ideas and get the basics down. Now I'm not going to go over the formulas and tell you how you can calculate things right now, but that's the idea behind the restrictive disorder. All of these is going to lower the lung compliance. All right. So next up, we wanted to talk a little bit about the second type of disorder, which is called obstructive disorder. Obstructive disorder. Uh, these are the problem with the flow. And sometimes this is also known as COPD, chronic obstructive pulmonary disease. Why is it obstructive? Because something is obstructed. We don't know why. Could be numerous problems. But if you have a flow problem, all right, that would be obstructive disorder. So for example, remember we discussed the trachea right here, or bronchiole, for example, and let's say there is some tumor in here. This tumor in here is blocking the airway. All right, now this is a lower amount of flow of air going in and out for whatever reason. All right, So this is a flow problem, it's obstructive. People with asthma. People with asthma, they produce a lot of the mucus, all right? There's gonna be a lot of mucus being produced right here, all right? With those goblet cells, if you recall, they produce a lot of mucus. Now look at the airway. The airway is not much. This is the reason why people with asthma attack, or when they get nervous or at times, you know, they have a difficulty breathing because there's not much flow going in and out. What do they use? They use bronchodilators, right? They have like this medication that, they put it in their mouth or their nose, and what it does, it goes into the trachea and into the lungs, and it opens up the bronchiole. So if you have the bronchiole now, it's more wider due to the diameter, even though there is a excessive amount of mucus, but it's still now the airway is sufficient enough for the flow of the air to go in or out. So that's the idea behind the flow problem or obstructive diseases. All right. So uh, a lot of times when you're looking at, let me go ahead and erase this. A lot of times when you're looking at sympathetic activity, you have the bronchiole, right? And then the bronchiole leads to the alveoli. There are sympathetic nerve fibers. The sympathetic nerve fibers, we discussed this, they release the, epinephrine, and as a result of that, what happens? Well, the bronchiole dilate, and as a result of that, now it's supposed to have more air going in and out, especially when you're exercising, when you see a bear, for example, your sympathetic fibers kick in, the wild animal is about to come in, and so on. So these fibers release this epinephrine, it dilates the airway, and it makes perfect sense because now you get more flow. More flow, that means more oxygen go into the capillaries that were surrounding in here. And we went over it already. So now what? Now there is more oxygen in the blood. There is more oxygen can get to the tissues, including the muscles. Now you can take off. All right. You have more chances of, in this case, overcoming the bear. Next. All right, so that is the what happens on the sympathetic activity, and the parasympathetic would be the opposite. So now that you learn about some of the disorders, and you also learn about the physical properties and the idea of ventilation. When we say ventilation, ventilation means breathing. All right. So hypoventilation and hyperventilation is the terminology. Like when you're breathing fast, it's hyperventilation. Hypoventilation when your breathing rate is lower. So therefore, it really depends on the state that you're in. Generally, we breathe about 12 to 14 times a minute. That's a normal, all right? But again, it varies from individual to individual, and it varies from your age, depending on your age, so you could have various breathing rates. And we are different with one another. But on the average, I told you what it is. Now that you understand this part, I wanted to talk a little bit about the idea of the generally the idea of breathing. A lot of times when you're reading the book, it tells you, okay, well, carbon dioxide is not good. You have to get rid of it. And the oxygen, we need to get into the cells and so on. But they don't tell you exactly why. So if we have a cell, all right, and looking at the overall picture, our cell can produce carbon dioxide. All right, this is a byproduct of the mitochondria. If you remember, mitochondria was a place for cellular respiration, and we mentioned that due to making of the ATPs and so on, the byproduct was carbon dioxide and water and such. So this is a volatile acid. What is volatile acid as opposed to non-volatile acid? Now, a lot of time hydrogen ion also need to be getting rid of the cell. As you know, hydrogen ion or contribute to the acidifying things, but you don't want your cell to get acidic because it can denature or it could affect the enzyme, it could affect the protein shape, it could do a lot of damages, it could wear out the cell membrane. So it's better to get rid of it and deal with it and so on. So this is a non-volatile group. So the volatile acid means that they don't get dissolved in a solution very well. All right. So if you go under the water and you blow air, when you blow air, that's carbon dioxide. What do you see? You see those bubbles that start forming from from in here. Or if you go ahead and you have the water in the beaker and you have maybe a straw and you blow carbon dioxide in here, what happens is these bubbles start forming. You could see that. All right. That means it's a volatile acid. Otherwise, if you wouldn't see anything, it would just get dissolved and that's it. All right. But you see these bubbles start forming. However, if you put in, you know, hydrochloric acid or vinegar in here, you notice this bubble is not forming. All right. Why? Because it's dissolving in here. Hydrogen ion can be dissolved in here, no problem. All right. So why are we caring about this? Because when your cells releasing the carbon dioxide, you don't want to create these bubbles because you have higher amount of carbon dioxide in your partial pressure. And during the capillaries that we discussed, all right, the capillary would have lower amount of carbon dioxide. So carbon dioxide can go from, for example, your cells or your tissues into the blood vessels because it's going high to low. But if it's creating these bubbles, wouldn't that be a problem? Because these bubbles can, get clogged up in here and interfere with the normal flow of the, you know, uh, blood. Also, if you inject an animal, let's say a frog, with air inside or carbon dioxide inside, they will die because again, these bubbles, it's hindering the flow. All right. And that's a big problem, obviously. Now, inside of this cell, we wanted the pH to be about 7.73, and outside of the cell, we want it to be about 7.37 to 7.43, which we can average about 7.4. Okay, that's our mission. So even though the hydrogen ion is going out, all right, we got to deal with it so that way we don't run into a problem. So for that, you need to understand a very important equation. And that equation, if you recall, was as follows. The equation was carbon dioxide combined with water gives you, by the way, this can go both ways, H2CO3, which is known as carbonic acid. And this carbonic acid can give you HCO3 minus, which is your buffer. And this buffer is also known as bicarbonate. And this bicarbonate or the buffer, you need to understand that the buffer resists a change in pH. Again, go back and review. Don't just like you memorize few terms. Find out what that means. And then also, this can go both, I'm sorry, that's it. So, and then what we also wanted to think about is the enzyme that can carry out this reaction happen to be the carbonic anhydrases. I just put Ca in here and Ca in here. That means Ca means carbonic anhydrase, and the carbonic anhydrase is an enzyme that can carry out this reaction. All right, so let's go ahead and analyze this. If you have higher amount of carbon dioxide and there's plenty of water, then the reaction is going to shift this way. All right, so of course this one is plus hydrogen ion. Sorry. All right, so the reaction is going to get towards the right in this case. But remember that this enzyme can go and catalyze this reaction either way. So if we have more of this part right over here, then the reaction can go the other way. All right, so it really depends on which way we're going to go into and whatnot. All right, and it gives you an idea. However, if we introduce just one carbonic acid in this equation that are equal, let's say you don't have too much carbon dioxide and you don't have too much bicarbonate, it's just like that. But what if we introduce one? What's going to happen? Is it going to go to the right or is it going to go to the left? So this reaction is in a way have a tendency to go towards the uh, carbon dioxide side. All right, that's the tendency if they have equal amount. But for the most part, that is the idea. If you have higher carbon dioxide and water is going to go the other way. All right, especially if you have this enzyme, this happens quicker. All right, so this is pretty important. Now, the idea of all that is, remember, our cells produce carbon dioxide in the previous slide I showed you. Outside of the cell, there is plenty of water. So now they can make a carbonic acid. Carbonic acid is a weak acid, all right? They can further dissociate if they have the enzyme available. And now you're gonna get a buffer plus hydrogen ion. Buffer, as you know, it can resist a change in pH, and hydrogen ion, we can deal with it. It can combine with some other things to form other things, and it could be dealt with different ways, as we've seen it in urinary system and whatnot as well, like for example, secretion. Therefore, you converted, or you have what you wanted to do is you wanted to convert this carbon dioxide that was volatile acid into something that is not, so you could deal with it, so you don't create a bubble, so that the flow of the blood is not going to be interrupted. So you're just changing a molecule to another, but now it's easier to deal with. All right, and this is the idea behind it. So I think on the one of the videos I told you guys, like, it's like a transformers, when the robots transform to a car or a vehicle or I don't know, the airplane and so on, they get to go a different route or faster, or they could fly, for example, and so on, which they were not able to do that before. All right, so it's kind of like that. And then once they get to their destination, they can transform back, right, to the robot, for example, and talk and do other things. Same thing with the carbon dioxide. Once they get to the destination, they can go in reverse. And then again, that's the time that you are actually in the lungs. That's the time that you wanted to breathe out the carbon dioxide, all right? So in terms of how do you deal with transporting, and how do you deal with the exchange of that, we can elaborate more on that. But before we go on, we could go ahead and do a simple experiment at home, and you can do this As well, if you have a pH paper, you could grab the water. The water is pH is 7. All right, so this is the water. Now, simply, if you put the straw and you blow air in it, all right, the air that you blow in is a carbon dioxide. So what is happening? Carbon dioxide is combining with water. And pretty soon you're going to convert and you're going to get a carbonic acid. And again, uh, you know, if you measure the pH, you will see that it's lower. Why is it lower? Because it's more acidic. So you just converted carbon dioxide and water to the carbonic acid. And plus, if you have the enzyme or if you leave it enough, it might dissociate to the buffer and bicarbonate and hydrogen ion as well. All right. So that's one of the things that you could do. For 2 minutes, you have to kind of like blow carbon dioxide in it, and then you'll be able to figure it out. In the laboratory, you have a pH meter, and you could just look at the pH meter. Initially 7, then after 2 minutes, maybe it will be lower. So lower than 7. All right. Let's go ahead and take a look at another experiment that you could do this at home too. Let's see if we can go ahead and demonstrate that. You can grab a glass of lemon juice and water. So put water and put lemon juice in here. Lemon juice is acidic. The pH, I don't know what would be the pH, but it's a low pH. That means it has a hydrogen ion in here. And then what you wanted to do, add baking soda. Baking soda is NaHCO3. All right, so in a solution, they will become Na plus and HCO3 minus, and that would be the source of bicarbonate. All right, so as soon as you add this, you see there are bubbles that start forming. All right, so what you're doing, you're adding these two, and then you're getting what? You're getting a carbon dioxide because this reaction is going towards this side now. All right, and these bubbles are containing the carbon dioxide. In fact, if you place a candle on the top and light it up, all right, as soon as you put in the baking soda, what happens to the candle? Candle requires oxygen to burn, right? So, boom, they go out. Okay, and you could do that in a family member. I remember 2 years ago, I showed this to my kids, and they kind of like really, were interested, and they say, "Wow," and I told them, "Oh, look, it's a magic. I could do this." But then I told them, but I'm not sure if they understood what I was talking about. Anyways, so that's what happens. All right, the candle is gonna go out. Now there's a lot of mosquitoes, and mosquitoes, they attract to the carbon dioxide, especially from your body that is emitting. So they come in and bite you and whatnot. You could look it up in YouTube, but there are ways that you could, kind of like build your own mosquito catcher, and you could apply lemon juice and some baking soda to produce the carbon dioxide. And I think there's like some kind of a funnel like this, all right, that you could place. And over here, you have to block it. So the carbon dioxide comes out here. The mosquitoes wanted that carbon dioxide thinking it's food. So they're going in there, and then they're going to have a hard time coming out and finding this hole in the metal. So they're going to have a hard time figuring out how to get out. So you could trap a lot of them as well. In India and other places with a lot of mosquitoes, infestation, tropical areas and so on, they use a similar technique in order to get rid of the mosquitoes. But they can carry diseases and such. If you like, you could go to the YouTube videos and explore some of those things and, make a project for yourself if you like. All right. So this idea is important and this equation is important because what we're going to be doing is looking at a transport of gases now. In other words, we wanted to find out how does the oxygen get transported, how does the carbon dioxide gets transported. Yes, the diffusion is in place. We know that already, okay? But how exactly does that take place? And, and we can go ahead and take a look at that, okay? So majority of the oxygen are being transported by way of hemoglobin. So here you can see uh, each hemoglobin can carry out 4 oxygen molecules, all right, and a single red blood cell. This is a red blood cell, and this happens to be the probably capillary, all right. So uh, from the lungs, right here is the alveoli. You have higher amount of oxygen. In here, you have low amount of oxygen. So oxygen will go right in there. These hemoglobin, they have attraction for the oxygen, and they get loaded, and basically now you're able to carry. So uh, approximately, majority or 90 plus uh, percentage of the uh, oxygen is being carried by via the hemoglobin. All right. So it's pretty straightforward. When you're coming towards the tissue, opposite will happen. In other words, now you have a lot of oxygen, all right, in the tissue, in the capillary, and then in the tissues you have low amount of oxygen. So basically, it's going to look like this. All right, so here's the alveolar. You can see the oxygen, it's 104 in this case, all right? And the reason is when you're coming, the uh, exhaled air, gas exchange, so you could see the partial pressure. So oxygen is 104, okay? But now oxygen is going in because there is a high level of oxygen. Inhaled air has a higher partial pressure of oxygen, so oxygen is going to go right in to the 104 and such, or from a high to the area that is low, so oxygen is diffusing in. All right. Now it goes to the lungs, it goes to the heart. Remember, respiratory system and the cardiovascular system, they work cooperatively with one another. And then from there, the heart is going to contract and send this to the systemic circulation. And once you get towards the tissue, again, you have higher amount of oxygen. And in the tissue, you have lower amount of oxygen. So again, it's no brainer. You have oxygen diffusing. As you know, the oxygens are also non-polar, so they have no trouble but passing the, the capillaries cell walls, and then the tissue cell membrane, and then getting to the cell and provide the oxygen that they need. So you could see what is happening. In terms of carbon dioxide, yes, they do go in from high to low now, and then going towards the right side of the heart, and then pumping towards the alveoli, and then again, same thing. You have higher amount of carbon dioxide in here, so it needs to go out, and that's what it does. All right, carbon dioxide exit from, again, higher amount towards the outside. However, they're not going to be dissolved in the blood vessels. So we need to talk a little bit about that, all right? And it requires that equation that we discussed. So in terms of the carbon dioxide, transport, we need to dig a little bit more. Let's go ahead and take a look at what is happening in terms of the carbon dioxide transport. All right. So 7 to 10% of the carbon dioxide is going to get dissolved in the plasma. The plasma is the watery area of our blood. All right. 20% is carried by the hemoglobin. All right. And we call it a carbaminohemoglobin. Whenever the carbon dioxide is attached to the hemoglobin, it's called carbaminohemoglobin. Whenever oxygen is attached to the hemoglobin, we call it oxyhemoglobin. Whenever the oxygen is out and dumped or unloaded, and the hemoglobin is by itself, we call it deoxyhemoglobin. So bicarbonate ion in the plasma, 70% is transported as bicarbonate. HCO3 transformers. So I'll go to the detail of this shortly, but this is the equation that we discussed. You need to understand what's going on, all right? And now we are ready to discuss this part. So take a look at what is happening. On this side, you have the tissues, all right? Here's the interstitial fluid outside the cell. It has fluid. And here is the capillary that I draw for you guys. All right. And here's the one red blood cells. Just for now, we are looking at it. All right. So again, we mentioned that a majority of the oxygen is being transported, loaded and unloaded via the hemoglobin. And you can see, look at the size of this arrow. Look at the size of this arrow right here. A thicker arrow means more. Look at the size. It's only O2 dissolved in the plasma. All right, very little. All right, very, very little. Okay. So the reason why it's thicker is because, greater amount, approximately about 98.5% of the oxygen is get carried out this way. All right, so they are attached to the hemoglobin. HB is for the hemoglobin. All right, so when they are unloaded, now the hemoglobin is available, but the oxygen is not. Why not? Because they got transported. So it's kind of like a taxi cab. An Uber driver, let's say they can pick up 4 passengers at a time, all right, and then go from one point to another. In this case, we are going from the lungs to the tissues. This is the tissues, that's our destination, all right. And you are unloading, let's say you're an Uber driver, and okay, we're here, you could get off now. 4 passengers in your machine or in your car is going to exit and go to their destination, in this case, tissues. It's kind of like that. All right, now you have availability. All right, now you could pick up other passengers, right? Okay, so this is kind of like a hemoglobin by itself. It can pick up 4 oxygen now, but it needs to go where the passengers are. Again, near the lungs. That's where you pick up the oxygen, right? Because the lungs, the alveoli are also very thin, and the oxygen that coming in from the lungs, it enters the blood that way. All right, so here we go ahead and take a look at maybe I can get this. The next slide, maybe this will be able to show us. Let me see. Okay, there it is. So now you could see it's from the alveoli or alveolus. Now you could see a lot of oxygen is coming in and they're getting loaded. All right. So again, very little, 1.5% dissolved in the plasma. The rest, 98.5, combined with the hemoglobin, and they're going to the destination. What is the destination? Well, I just told you, when we get to the tissues. So that was how you transport oxygen. What about the carbon dioxide now? Yes, tissues producing a lot of carbon dioxide. All these carbon dioxide need to be exiting, and you don't want to create bubble inside. So look, that's why 10%,
Out of these 10%, 7% of it dissolve in the plasma, right? When you blow carbon dioxide inside of the water, a beaker of water or glass of water, you see a lot of bubbles, but a very little bit it gets in the liquid, it gets dissolved, not a whole lot. All right. So 7% right over here. Another 3% is going to combine with water and make the bicarbonate and hydrogen ion. All right. But this process is slow. Why is it slow? Because we don't have that many enzyme in here to carry out this reaction fast. So if this is maybe 7%, this would be 3%. Again, the size of this arrow is telling you kind of like the stories and such. All right. Now that we used up about 10%, we have 90% of the carbon dioxide available. What happens then? 90% of the carbon dioxide, they're going to go to the red blood cells. They are nonpolar, they can pass the membrane, there's no problem. And now what happens? 20% of that 90% is going to combine with the hemoglobin. Remember, they are available because they dumped the oxygen already. So now they could pick up another passengers, in this case, carbon dioxide. And when does that happen? That's called a carbaminohemoglobin. That's what it is. But the majority of it is turning to totally different entity. From carbon dioxide that combines with water, our red blood cells have carbonic anhydrase. And that is allowing it to convert it to a totally different molecule, which is bicarbonate plus hydrogen ion. Now, the bicarbonate can go and exit out, and instead chloride is going to come in. They're both negatively charged, so in terms of electron neutrality, you're not going to mess up with the cell negativity or positivity. All right, one negative goes out, one negative comes in, and this is called a chloride shift. All right, but what just happened? Well, first of all, we got rid of the bubbles now because they don't cause bubbles when they are not carbon dioxide. Now they are totally different entity. Another thing that is happening now, bicarbonate, which was a buffer and buffer resists to a change of the pH, all right, is in the plasma. And this outside is the plasma, all right? And this plasma, what happens now, it can resist a change in pH. So if you're eating something acidic or whatnot, to a certain extent, it could tolerate that because of this buffer. Some of these hydrogen ion that uh, is remaining, again, they can combine with the hemoglobin for the time being, all right? But if it's way too much, then that creates problem, but you don't need to be too much because it's 1-to-1 anyway, so they are attaching to the hemoglobin for now. That is, again, is not causing problem. Remember, on the plasma, there are a lot of protein molecules, there are other stuff. When you're introducing a lot of hydrogen ion, they can interfere with the normal shape and such. So if they are contained, attached to the hemoglobin, they're not going anywhere, then you're okay. All right. So one more time, you have 100% carbon dioxide that is releasing from the tissues. 10% of it goes into the plasma. Out of that 10%, 7% of it dissolves. 3% or so combined with water, they become different entity. All right, but again, this process is not very efficient because you're lacking the enzyme, but you do have the enzyme inside of the red blood cells. So 90% of the carbon dioxide is going to go in, which 70% of that 90% is going to turn into the bicarbonate and hydrogen ion, and 20% is going to combine with the hemoglobin. All right, so now what? Now you contain majority of the carbon dioxide, it's in the red blood cells or being transformed to another type of a molecule that does not cause volatility or bubbles. Now what? Now you're able to transfer them without any problem. Well, where they are going? Remember, respiratory system work cooperatively with the cardiovascular system. So if we go ahead and take a look at in here, when you loaded the carbon dioxide, where are they going? They're going towards the heart right here. So what? So they could be pumped. All right. So they could be pumped towards the lungs because we were right here. We were at the tissue. All right. We were at the tissue. We need to get to the lungs. Why? So we could expel the carbon dioxide. All right. So uh, that's where this slide comes in. All right. So everything is in reverse. In other words, now bicarbonate moves in and chloride goes out. That's a chloride shift again. It's a reverse chloride shift. And now they can combine with the hydrogen ion that was carried by the hemoglobin. So because they have the carbonic anhydrates, this is pretty effective and fast. So what happens is you're making the carbonic acid. After that, it dissociates to hydrogen and carbon dioxide. Carbon dioxide, again, majority of it, 70% or so, it goes into the alveoli, and you get to get rid of it via the exhale. On the other hand, about 20% of the carbon dioxide was combined with the hemoglobin. They get separated, they're time to be unloaded, and that's what happens in here. And again, about 7% was dissolved in the plasma. They reach in here. Again, all of these, what does that tell you? All in here, is a high carbon dioxide. In alveoli, you have a low carbon dioxide. Yes, this is a diffusion process that you're familiar with. The only thing is happening, you're going back. If earlier, if the robots were turning into a vehicle, for example, to be able to transport faster and more efficiently, now the robots is turning into the vehicle is turning back to the robots, and that's what is happening right now. Okay, so that's the idea about the transport of oxygen and transport of the carbon dioxide. All right. Uh, Next up, what I like to do is take a look at, okay, now you might say, okay, well, we did the transport. Now what are we going to do? In other words, we need to go back and take a look at how we can regulate things and how we can control things. All right? In other words, why is it that when we are exercising, your respiratory rate increases, and opposite happens when you are in, rest? So for that, we need to take a look at the breathing control mechanism, which has a little bit of physiology behind it. Okay. All right. So let's go ahead and take a look. First of all, you have these chemoreceptors. Where are the chemoreceptors are located? There are two kinds of chemoreceptors. The chemoreceptors happen to be on the carotid arteries as well as in the aorta. So same places that you had the baroreceptors, you could see these chemoreceptors. You will see these chemoreceptors. All right. So there are sensors there. What do they sense? Well, in general, before I go ahead and talk to you about, there is another type of, now these are called peripheral chemoreceptors. So these are peripheral chemoreceptors. Why is it called peripheral? It's called peripheral because it's outside of the central nervous system. Now, there are some chemoreceptors within the central nervous system, in particular on the medulla oblongata, right outside of it, where they are sensing the cerebrospinal fluid. And these are called a central chemoreceptor. In general, these chemoreceptors, they detect higher amount of carbon dioxide, low amount of oxygen, and a higher amount of pH. I'm sorry, hydrogen ion, or lower amount of pH. That's what they are sensing. But majority of these chemoreceptors, in terms of sensitivity, they sense increasing carbon dioxide. All right. So Earlier in the semester, we discussed that you had a receptor via the sensory neuron. You had the control center or central processor, and then you had the motor neuron, and then you had the effect. We understood this part in the nervous system, we understood this part in the simple reflex physiology, we understood this part in the homeostasis. That's pretty much the idea. Well, in terms of the receptor, you have chemoreceptor. In this case, majority of it, let's say they detect carbon dioxide. Sensory neuron senses changes in the blood, for example, because they happen to be either in cerebrospinal fluid near the central chemoreceptor or in the blood. These chemicals, carbon dioxide, increased amount of carbon dioxide. This happened to be in the aorta and the carotid arteries, as I showed you. Anyways, they detect that. Okay, they're sending via the sensory neuron to the control center. Where is the control center? The two control centers for breathing happen to be pons and medulla. In this case, it's in the brain. Pons and to the most expensive medulla oblongata. Medulla and oblongata, majority of the majority of the control centers are there. Well, you might say, wait a minute, I thought the receptors were there. Yes, the receptors are there, but they can sense things and bring in information to the control center, to the brain, inside of the medulla. Those receptors happen to be outside. All right, so now what? Now, via the motor neuron, information is going to exit and reaching the effector. Effectors were muscles and glands. So, if we go ahead and take a look at, we already see some of the muscles and glands. Diaphragm, here are the other effectors, intercostal muscles, and that's what we discussed. We can see the sensory neurons right over here. These are the sensory neuron, and again, arrow coming out is the motor neuron. So we got all the components. You got right here, it says breathing control center. All right, so we got the control center. So we got all the components already. Now we got to put it together. All right, so let's think about this for a moment. Let's say that you have let's say that you stop breathing for a moment. You could hold your breath. When you're holding your breath, what are you doing? You are doing hypoventilation. When you are not breathing. Of course, you're going to die if you don't breathe. We're just saying, like, let's say 30, 40 seconds, however many you can, right? You hold your breath. So if you're not expelling the carbon dioxide in your blood, the carbon dioxide is going to rise right here. And the chemoreceptors, the peripheral and the central, they're going to detect this carbon dioxide, right? Via the sensory neuron, the control center is going to be notified. Via the motor neuron, they're going to stimulate the muscles. So after the 30 seconds is over, what do you see? Your breathing rate is much higher because of that, all right? Because you want to get rid of this carbon dioxide, all right? Carbon dioxide was high, you want to get rid of it so it be low. So your breathing rate along the way has to be increased and make sense, because when your breathing rate is high, you're getting rid of the higher amount of carbon dioxide out to balance. This is not going to continue forever. Once you reach the balance, your breathing rate also balances, and that's it. That's the end of it. So if you already hold your breath, give you a little bit of time for you to notice how fast you're breathing at this time. All right, by now you probably have to let go and think about it. Think about what just happened. All right, so again, that's what is happening. Okay. So, uh, what happens during the hyperventilation then? During hyperventilation, now is the time that you wanted to breathe faster. This is not very healthy, so if you want to do this, don't do it more than, like, 7 to 10 seconds. All right, but when you're hyperventilating, you're getting rid of the carbon dioxide and you bring in more oxygen. Remember, the idea is more is not better. All right, the idea is balance is the best. All right, so during hyperventilation, when you're putting so much oxygen, you're getting rid of the carbon dioxide again, you're messing up with acid base, you're messing up with the amount of gases and so on. So you could actually faint if you cannot tolerate this, and it's not healthy to do. But a lot of divers, to hold their breath for a longer period of time, they hyperventilate before they dive, especially if they don't have oxygen tanks and so on. And some people enjoy going deeper, and they hold their breath, so they hyperventilate. And again, by doing that so many times and so on, they're able to dive further. And again, the experiment, if you involve in how does the seal, breathe longer period of time and so on, discuss these ideas a little bit further. All right, but when you're hyperventilate, you bring in more oxygen in your system, so theoretically, you'll have more oxygen in your blood. And now, when you're holding your breath, those oxygen can still help you out in terms of your normal functioning of the cells, and that's what happens. Okay. Now, how do we know that we have the normal rate, normal amount of air in our lungs? Our lungs is healthy, our lungs can expand, our lungs have no problem with restrictive or obstructive and so on. Well, there is a device known as a spirometer. All right, so the spirometer is a device, or also known as the spirometer. The spirometer. Uh, yeah, or another name, pulmonary function test. All right, or PFT, pulmonary function test. All right, so pulmonary function test or a spirometer, it can measure the lung volume and also the capacity. All right, so it can pick up potential pulmonary disorders. All right, and give you an idea about how well your gas exchange is taking place. All right, so for that, we could go ahead and take a look at the device, a spirometer. All right, and you could go ahead and take a look to figure out what is the problem. And in order to understand this, we need to revisit the ideas that we learn about obstructive and restrictive. So let's go ahead and pretend that we have that device or apparatus with us, and you are measuring the lung volume. So these are some of the graphs that are associated with using the spirometer. Normally, and again on the next slide, in which you can go ahead and use that as a reference, it gives you the explanation on each one is color coded. So tidal volume is just normal quiet breathing. So if you see a lot of these and so on, you should be able to count, like, if the x-axis happens to be the time, you should be counting like around 12 to 14 of these in 1 minute. All right, so when it goes up, again, the volume is increased, so that would be inhalation normally. When it goes down, is exhalation, inhalation, exhalation, inhalation, exhalation, inhalation, exhalation. Depends how many times you're breathing. If you're breathing more and rapidly, then this would be closer to one another. If you're breathing like, less and so on, this would be taking longer time, okay? But this is normal amount of air that is not forcefully, just quietly going in and out of our lungs. It's called tidal volume, also abbreviated as a TV. All right. But what if you wanted to forcefully inhale or forcefully exhale? Well, normally you exhale and then forceful exhalation, you could measure it. So in other words, the spirometer is a device that have a tube. So first you could normally exhale and then forcefully exhale in here. And that gives you the ER value. ER value is for expiratory reserve volume and basically is forced exhalation after normal, quiet exhalation. That difference is called ERV. Still, if you get all the air out of your lungs as much as possible, you exhale, still there are some air trapped in your lung, and that's called the residual volume. Residual volume you cannot measure, you could just estimate. What about the inspiratory reserve volume? Again, normally you inhale a certain amount of air, but if you forcefully inhale air after that, that's called IRV. All right. But perhaps one of the major things that physicians do to find out if you have a healthy lungs or whatnot is to measure the vital capacity. Vital capacity, you could look at it in the Google for your age, for your height, but three things are important. First is your height, then is the age, and then it also your sex is also important. Right? Generally, male is larger individual, so the lung volume or the vital capacity for the male expect to be higher. So this number might be higher. Age is also important. As you growing, your lungs is becoming bigger. As you growing way older, there is a lot of ossification that we discussed, deterioration of the lungs, so your lungs not going to expand as much, and that will affect it. Height, I mean, if you're a larger individual, taller individual, then you probably will have a bigger lungs. And versus if you are a petite individual or shorter individual, you'll have a smaller lungs. So they can have a different capacity in there. All right. Now, therefore, you could look this up. Just like, we said 120 over 80 is supposed to be a normal blood pressure. Well, this is different now. This number is different. If you're a younger individual, have a healthier young lungs, that might be different. It might be higher. If you're a taller individual, your lung might be. So it's not one number general for all. It really depends. So you really have to look this up. You could just Google it. What is my vital capacity, for example, and indicate your height, age, and sex, and it will give you a number. If you put in a different one, it will give you a different number. And those are the standards that you could always take a look. All right. But here's the idea behind all these tests that we wanted to do, and why is it important to pay attention on this one. Let's just give you one example and then we'll carry on. Earlier, I told you about different type of lung problem. All right. So to name a few, we mentioned that some of those were the asthma, for example. Right, here's the asthma. You can see the mucus is in there and is obstructing it. All right. Now, let's say you want to get the vital capacity. What is vital capacity? Yes, it depends on age, sex, and height. But how do you get it? Here's how you get it. You get the spirometer, which is apparatus and has a tube where you're supposed to blow air in it. All right. So before you blow air in it, you inhale air as much as possible. All right. When you inhale as much as possible, your lungs is going to get expanded as much as possible. Then you exhale as much as possible. All right. And then the recording in here, it shows you what would be that number, let's say 4,000 milliliter. All right. Well, if you have problems in the lung, let's say, airway is blocked. Now, not the same amount of air as in here. Remember the flow that we discussed is different than here. Same thing with the cystic fibrosis. All right. Same thing with inflammation or bronchitis. All right. So look at the diameter in here. Look at the diameter. They all have lower amount of flow. Here is the emphysema where the air is trapped in the lungs. People sometimes refer this to as barrel chest, all right? Because it's kind of like you have a barrel, the way that your chest is forward or whatnot. Person is not showing off. It's just, uh, you know, a lot of these alveoli losing their surface areas and excessive amount of air is trapped in the lungs. All right. So they are overstretched and ruptured, and unfortunately, these are a lot of large amount of space of uh, less surface area for the exchange can be offered at this point in time. Anyways, all of that will show on the vital capacity because when you inhale as much as possible and exhale as much as possible, blow into the device, if there is less air going to your lungs, less expansion. And either, or if you have deterioration of the lungs, again, whether it's restrictive or obstructive, you'll get low vital capacity. And then further diagnostic can be taking place, and you should uh, you know, look to see why it's so. Is it a genetic problem? Is it, infection problem? As you know, when you are getting infected, like chronic bronchitis, there is an excessive mucus production, just like asthma. So it's hindering the opening, all right? This person is coughing, is infected, the lung is irritated. A lot of times, tobacco smoke and the air pollution cause these underlying conditions, and now coronaviruses too, all right? So they break down the connective tissues of the lung. They could lead to the emphysema. People that are the smokers, they could have a higher percentage of lung cancer, bronchitis, all right? The airway can get obstructed, all right? This could lead to other problems such as frequent infection, hypoventilation, hypoxia, which is lower availability of oxygen, respiratory acidosis. Now the acid is and base is not going to be monitored as well. All right. So all of those could be potential problems. All right. So looking at the highlights of what we discussed, it looks like we looked at the pressure and the volume, Boyle's law. We looked at the structures that were important. We looked at compliance, elasticity, surface tension. Remember the surfactant cells. We discussed the gas exchange in the lungs, both oxygen and carbon dioxide. So we discussed the regulation of the breathing, sensors, peripheral and central chemoreceptors, what they do, what do they sense, and how they monitor increasing the effectors or respiratory muscles, for instance. We discussed the gas transport. Carbon dioxide in particular was a little bit more detailed explained and such because the carbon dioxide, how they get transported is not just in the plasma or combined with the hemoglobin, like 98.5% that happened via the oxygen. All right, we looked at the chloride shift and how carbon dioxide needs to be changed into the bicarbonate and hydrogen ion to be transported effectively without forming any bubbles because they are volatile. And the acid and base, if you don't deal with the hydrogen ion, you're in trouble. Right? That's why the hydrogen ion combines with the hemoglobin. That's why it can combine with the bicarbonate when convert to the carbon dioxide, so we could expel them and we can deal with these hydrogen ions so they don't denature our protein inside of our cell. They don't mess up our enzyme, and that is conclude our discussion for this chapter at this point in time. I hope you learned something and you enjoyed the lecture. And good idea not to smoke, good idea to not get near a person that is infected because chances are you might get infected. Better to not inhale as much as possible, the pollutants, dust particles, things that are around us. Now with the availability of the face mask and so on, might be more beneficial at times, all right, to prevent yourself from getting infected from various diseases. It's not just, uh, you know, coronavirus. It's many, many diseases. They are airborne. They go through your respiratory system. And once they get in the lungs, the conditions are ideal for them to reproduce. It's the lung uh, has a nice temperature, ideal for bacterial growth, other microorganisms, viruses, things in that nature. There are plenty of gases, including the oxygen there for them as well. So they can do a lot of damage there. Anyways, be safe. Hope that your lungs also will be safe.